Y bueno, yo tengo el gusto de presentar al Dr. Helmut. First of all, thank you very much for coming to share your research experience. Y bueno, el Dr. Helmut hizo su postdoctorado en el Departamento de Medicina Metabolomica y Nutricional en Múnich, donde actualmente él trabaja y se desempeña como investigador. Él tiene mucha experiencia en el área de metabolómica y bueno, lo que nos va a presentar hoy está relacionado con la programación fetal. ¿Cómo es que el ambiente durante la etapa gestacional influye en la salud del individuo? Entonces nos va a hablar un poco más basado en la dieta, qué tanto la dieta va a repercutir en la salud en la edad adulta. Y bueno, todo esto está relacionado con los cambios epigenéticos. Entonces, bueno, démosle la bienvenida al Dr. Helmut. Herzlich willkommen.
So, I first have to thank, of course, for the kind introduction and for the invitation to speak here and for all the organizing of this trip. I also thank the Institute and the University for the invitation here and to present some things about early programming, and I will especially focus on metabolomics and just in a few slides on epigenetic styles. So I hope you had good rest and you got set all these epigenetic mechanisms, and I will now present to you the results of our group. So first, where I'm from. This is Europe, you maybe know, and in the middle of Europe there's Germany. So I was born somewhere here, but now I live in Munich and I work at the LMU, the Ludwig-Maximilians University of Munich. And there's another university, so I have to mention this. And our group is located at the university's hospital in the inner city. So there are two hospitals in Munich, one is in the outer city and one in the inner city. But the children's hospital is still in the inner city, and as you see, it's the hospital. Our targets are humans, and especially children and pregnant women. And this is our group, the Division of Metabolic and Nutritional Medicine, and it's a very big group, so it's not everyone on this picture, but not everyone is there all the time, and we are a very heterogeneous group, so we have statisticians, we have chemists, we have nutritionists. I'm a pharmacist, actually. We have a lot of pediatricians, and we are all supervised by Professor Berthold Koletzko, who's very famous for its early protein hypothesis, saying that an infant formula with higher protein fed to infants, they have a higher risk to become obese in later life. So this is our trip during my talk. So I first want to introduce early programming and why we do early programming and studying early programming. Then I will give a short introduction to metabolomics. If you have any more questions, you can of course ask me afterwards. And at the end, I will present the results in our early programming studies. So what is early programming? This is a review which was published last month, and this was a review looking at the body mass index and to look how obesity is developing in the last year. So there was really a huge review in 200 countries in a long period, so about 40 years, by the NCD risk factor collaboration. NCD means non-communicable diseases like obesity, diabetes, or cardiovascular diseases. And they compared the prevalence of obesity or of the body mass index in all countries in the 70s and in nowadays. So when you look at this map, you see the mean BMI. So BMI, body mass index, is a marker for obesity, and if you are obese, you have a body mass index higher 30. If you're overweight, you have a body mass index higher 25. And you see in the 17s, there is a lot of orange here, especially in the North American part, and there's also a lot of high or low, bright yellow parts here. That means there's an underweight population in Africa. But what is striking is that we here already have some dark orange. And dark orange, as you see here on the scale, it's around 27 mean BMI. That means in the mean, in the average, the North American is overweight. But when you look now at the map from the 2014, you see a lot of red. And red means it's over 30. And over 30 means obese. So in the mean, a North American is obese. And this is really dramatically increasing. And what they especially found when they looked at all these countries and at all these different developments in these countries, they found that nowadays more people are obese than underweight. So in the years before, in decades before, you had a lot of underweight prevalence, which you still have in Africa and South Asia. But obesity is becoming more and more and an increasing factor around the world. And they also said that in 2025, severe obesity, which is a BMI above 35, will surpass the underweight prevalence. So that means that in the complete world, there are more people severe obese than underweight, and this is really something we have to track and to tackle somehow. But in this review, they also stated that there's no intervention at the moment which is possible to tackle this increasing pandemic of obesity. And so this is where we, for instance, came up in our group to look how we can prevent or tackle the obesity problem. What is the problem with obesity? Obesity is a risk factor for all these non-communicable diseases like cardiovascular disease, kidney diseases, or diabetes, and even some cancers. And of course, they are related to morbidity and mortality. So if you have higher obese people, higher obese population, they are more likely to get one of those diseases, die, or get severe illness. And of course, it's not only bad for the humans, it's also bad for the socio-economic system. The route to obesity is very complicated, so it's not fully understood yet, I have to say. There are some genes which are related to obesity, so some genetic determinants. Of course, it's food. So, if you eat a lot of fat and bad sugars, you have a really high risk to become obese. But there's also physical activity, you have stress, even in the office, and smoking. It's all parameters which are related to obesity. There are a lot more environmental parameters which are related to obesity and may result in this obese population. Which you also have, or what you also have to be aware of are these different stages of doing your lifespan, your life cycle. So each of these factors actually can influence you differently in all these different stages of your life, and there are some plastic periods where your metabolism can be more influenced than in other parts of your life. And this is in, of course, pregnancy, it's the first months of life, in infancy, and during puberty. And there's another issue with obesity, it's the intergenerational cycle of obesity. That means if you have an obese woman, this obese woman gets pregnant, she will get an obese child, which is growing up and is becoming again an obese woman or obese male. And if the obese woman gets pregnant again, she gets again a pregnant child. So this is some kind of an intergenerational cycle, and it's very hard to break this cycle. So what was the early programming? As I mentioned, there are these flexible metabolism periods in life. So periods where your metabolism is not really fixed and where you can influence this metabolism by exposures, by environmental exposures. And then you can set your metabolism by these exposures. And when this is set, it's more or less set. And then you can become later obese, although it was in the first period of life. So what is the first period of life? It's pregnancy, birth, and infancy. So usually you say it's the first 1,000 days of life. That means pregnancy, 9 months, and the first 2 years of life. And there you have all these factors which may influence the development of your metabolism. And like nutrition is of course one of the most important factors, but you have a lot of more exposures like stress of the mother, placental development, you have obesity of the mother, you have diabetes of the mother, you even have some kinds of pollution. So there's a new review, I think it's yesterday it was published, where they just showed that some toxigens which you set up during this early period can influence your metabolism. And if you influence your metabolism in an adverse way, they can set it to something like, let's say, fat storage, and then you become later obese in later life. So since this early exposure of these factors influence your later life, this is what we call programming. I also have to mention that there is a developmental origin of health and disease, the Doward hypothesis, which is pretty much the same, and Barker et al. In 1990, who found out that low birth risk is a risk factor for later cardiovascular disease, and this is known, or it's famous to be one of the first programming paper published in this field. So what do we do to investigate this early programming period? So we have a lot of different cohorts in humans, so nearly all of them are humans. We have one animal model, it's a baboon animal model in our division, and we have a lot of cohorts or intervention trials. So if you look at these blue points, these are intervention trials where we looked if different intervention in pregnancy or in infancy can affect the later metabolism or the risk for obesity. We have a lot of blood in these cohorts, even longitudinal follow-up in pregnancy. Those are the red points here on the slides. And so, we really try to find results which exposure influence metabolism and then influence the risk for obesity in childhood or in adulthood. And what we do in our group is especially looking at metabolomics. And so I will now give you a brief introduction to what metabolomics is. Metabolomics, as the name said, is the determination of metabolites. So this is a very easy part. What is a metabolite? Gets a little bit more complicated because it's said it's a small molecule with a weight less than 1,500 kilodalton. Maybe this is a rule, but I'm not sure because 2 years ago it was 1,000 kilodalton. So they somehow always adapt this limit. So for me, a metabolite is a small molecule which is involved in a biological process. So it can be a substrate, an intermediate, or a product of a biological process, but it's not a protein because they're much more heavier. And so this is a metabolite. And usually you say metabolomics is the analyzing of all metabolites, and if you analyze the complete set of metabolites, you get the metabolome. The problem here is that, of course, we did not know any metabolite. There are so many metabolites. You can measure them all, even if you know them. And there are some metabolites which are so low concentrated you can't even find them in your tissue, in your cell, or in your body, or wherever you are looking at the metabolome. So usually the term metabolome is used by different groups just by the metabolites they measure. And you have to be very careful because different groups measure different metabolites, but they all refer to it as a metabolome. So why do we metabolomics? So this again is a genotype. So I said there are some genetic determinants for obesity, and the phenotype, which would be obesity. And then you have exposures, which influence the complete metabolism. So the genes can be affected, no, not so really affected, but the epigenome can be affected, the transcriptome can be affected, the proteome, of course. But if the epigenome is affected, also the transcriptome and proteome should be affected, and the metabolome. So the metabolome is a downstream product of all these other omics and of all exposures. So it's very close related to the phenotype. So if something is going on in the metabolism in one of these steps, or it's just initiated by the exposure, you have to see it in the metabolome. Of course, you have to measure the right metabolites. And so this metabolome is very dynamic to different stimuli. You can see if you have an intervention what is really going on in the metabolism. And yeah, this is why we do metabolomics. In usual, there are two different kinds of metabolomic studies. So the one is looking for biomarkers. I think this is the most famous one, where people look if different metabolites are related to obesity, insulin resistance, diabetes, cancer, and so on. They even look if there are some pre-onset of the disease, if there's higher or lower metabolites. So just to predict if someone gets a severe illness. But of course, there's the other thing to look or to get some ideas of what is going on in the metabolism if you develop a disease. So you have maybe different stages of your disease, and different parts are affected at different stages. And so if you analyze the metabolome before the onset of disease and during the onset of disease, you can get some idea what is really happening in the mechanistic way, in the pathology. And the main idea of this is to find targets for interventions, where you can say, okay, there are some changes in the lipid metabolism at a certain kind, and then you can adapt the diet of children or of pregnant women. So there, can you tackle this mechanism? So when you do metabolomics, at the end you get a lot of metabolites. So measurement is more the easy part, but then you get a lot of these metabolites. Each point is a metabolite. This is a famous Keck pathway map. It's very complicated. Maybe there are all metabolites we now know are in this map. What we measure in our division are these metabolites. So we focus a lot on lipids and amino acid metabolism. And usually we get a lot of concentrations for all these metabolites. And afterwards, we have to look what is really going on. And this is, you don't have to know the slide or remember the slide on any time, but just to show you how complicated it is. So these are all metabolites here. You have some enzymes maybe. And then you find alterations in your trial. And then you have to think of which pathway is affected. Does it make sense? And it's a very hard work to interpret all the data you have. So metabolomics and cohort studies. So this is one of my favorite slides, this first thing. When I started at our division to be more or less responsible for all the metabolomic studies, I created this slide because there were a lot of studies going on where they just said, okay, I want to do metabolomics. And we just did metabolomics. And the problem is if you do metabolomics, you don't have an hypothesis, you have no idea what you're actually doing. You measure all metabolites, you don't have an idea about the question or anything. So if you want to do metabolomics, you need some idea and not just do it because it's fancy. So afterwards, it's more easy for you to get a paper, so this is the main aim, of course, but even to get some idea or results out of this. And it's more easier for us to work with this, since if you don't have an hypothesis, you're really looking at everything and you have no idea, and you really got stuck up with all this big data. So what you should do before the determination of the metabolome is to create an hypothesis in the best way. You have to look at the samples you have and at the data you have. So if people want to do metabolomics, they just do it because they have some samples left. Metabolomics is never the first or the main outcome of a study. It's just a secondary outcome. So the blood samples are usually used for something else to analyze. And so you have to look what samples, how were they treated before, how much volume is left. And afterwards, because I just mentioned it because I had a lot of these cases, you have to look if the data fits to your samples. So if you want to, for instance, look if children are obese to a certain metabolite, you have to have obese data for these children. Sometimes partners send us samples, we analyze them, and at the end we just need 30% of our samples because they don't have the data. And this, of course, is a little bit annoying. Since for metabolomics you need a really, really high power, and studies are not set up for metabolomics. So the power is usually less than is needed, but you should do the best of this. So I always said prepare an outline, agreement, or plan, whatever, just to get an idea what to do and how to do it, and ensure funding. So people usually think you just do it in the mass spec and then you get all your data and you're done. But this is the cheapest part. The most expensive part in doing metabolomics is the manpower you need for the data analysis, statistical analysis, and interpretation. And just for you to be aware of this if you ever want to do metabolomics, because in our division, at the beginning, it was just calculated for the measurement, and then we had no funding for doing all the analysis, and this was really hard times. So, metabolomics and cohort studies. So, as I mentioned at the beginning, we are looking at humans, children, infants, and pregnant women. So, all these samples you see here on this slide, you will never get from these populations. Because of ethical reasons, of course, and you should not do it, of course. But in each of these tissues, something different is going on. And we just have blood. So blood, of course, is appreciated because it's not so invasive to sample. You just put a needle in someone. They have to agree. In children, even this is really difficult. And, of course, this is just a transport compartment. So if, for instance, for gluconeogenesis, the glucose is generated in the liver, and then it's transported via the blood to the muscle. So usually, liver gives glucose to the blood, and the blood gives glucose to the muscle. But you will not see it in the blood, because the glucose is just a transport compartment, and the glucose level in the blood is very stable. Even if gluconeogenesis is really activated, you cannot see it in the blood, and then you need tissues, and of course in our populations you don't have tissue. So we have no information about what is going on in the cell. This is why we have these large cohorts, a lot of large cohorts, where we have to repeat all these findings to create some evidence. What we do is targeted metabolomics. As you saw on the slide where I mentioned the metabolites, we know what we measure. There's the other part of metabolomics where you look at non-targeted metabolomics. That means groups who do non-targeted metabolomics, they actually have not so much idea what they measure. They just put their sample in their device, usually a mass spec or a NMR for a nuclear magnetic resonance spectroscopy. And then they scan everything they can find, and then they have to do a lot of data analysis work to normalize and to look if they have repeated findings. And this is a very complicated business, especially when it comes to the data and to analyze the data. So non-targeted analysis is usually done in very smaller groups. They don't have so much samples you have to correct for everything. So what we do in our group is target metabolomics. It means we know what we measure. So we, for instance, look at a lot of lipids and amino acids. And of course, the disadvantage is that we are limited to just the things we measure. But what we do is to look at all the literature and the work of other groups who perform untargeted and smaller samples to get an idea which metabolites may be interesting. And then we set up methods to analyze these metabolites. The advantage, of course, is that we can quantify, that we are specific, so we actually really know. We have standards, we have internal standards, which are necessary and expensive, but we can quantify and we know which metabolite it is. In an untargeted approach, you did not know it. You just get a peak or a signal for a mass spec, and then you have to somehow interpret it, and you have some idea which metabolite it can be. And it's more easier handling, especially when it comes to data analysis. So the usual equipment for a targeted metabolomics platform is a triple quadruple mass spectrum, where we have two. This is a more newer one, but it's actually also quite old now, but we get a new one now. And this is the older one. This is Dr. Olaf Uhr, who is responsible for the lab in running all the cohorts through the lab, because you can, as you imagine, it's very difficult to run such samples. So we have around 1,000 samples per cohort, and it's very difficult to ensure the quality in the complete measurement, and that's a stable signal for everything. So we measure amino acids because they're very high concentrated on a more or less sensitive equipment, and we have these other 4 applications on the more high sophisticated equipment. And what we need is just 50 microliter of plasma. These are our technicians, Alex and Steffi, who did all the work in the lab. Of course, this is also a lot to do, and now have established a kind of routine in our lab. I think this is quite new for Metabolomics Society's work, that we are now just doing routine work. So we just have this method where we use this 50 microliter of plasma. We dilute it with methanol to participate the proteins, which otherwise would disturb our measurements. And then we aliquot the supernatant of this into 4 the different methods. And it's really high throughput. So in our division, each day there are, I think, 100 to 200 samples, and it's really high throughput measured and done. And so we need a lot of manpower actually for this. And as I mentioned, we have a clinical target metabolomics, and we have these 5 different approaches. That means if you want to do metabolomics, for instance, with us or with other groups who have different approaches, you have to be aware that each of these methods is a single method. So it's not just one injection in the system, in a mass spec, and you get all your data. It's 5 injections. And so you have to be aware that each of these methods is a consumption of time and solvent. And as I mentioned before, and you see I'm very pointing at this, you have to generate a hypothesis before you start the laboratory work, because, for instance, when you look at the non-acidified fatty acids, these are free fatty acids which are released in a starving or fasted state. We have a lot of infancy trials. That means an infant usually is not fasted. You cannot do this with an infant because it wants to eat all the time. And so there's no sense in measuring this non-acidified fatty acids, since there's a big variation just by the fasting state. But at the beginning we also did it because we were not really aware of this, and this is a really expensive method, and so it's good to have an hypothesis before and to know what to measure, so you can skip time and solvent consumption steps. So this was the introduction to programming, to metabolomics, to our cohorts, and I will now show you some results which we found in our cohorts. The first one will be about pregnancy. So this is Karen Lindsay, Dr. Karen Lindsay from the University of Dublin, who stayed in Los Angeles to analyze together with us the metabolome of the UCI cohort. The UCI cohort was originally set up, of course not for metabolomics, it was set up to analyze how stress and diet affect the adiposity of the infant. And so they collected a lot of data about diet, about the mothers in pregnancy, so even in each trimester, and a lot of data about the children. And we first focused, as you see here on the left-hand side, on data in pregnancy. And we got, at the end, you see it here at the end, we got samples from around 130 women which gave blood in each of these trimesters. You see we excluded non-fasting. As I already mentioned, in the non-fasting states, metabolites are different, altered than in the fasting states. So usually these samples would somehow disturb our analysis. And what we also did in this trial was to exclude diabetic patients from this study, since it's very few numbers, so it was a very healthy population when it comes to diabetes. And we could not run a single analysis for diabetes by this small sample number, and so we excluded them. And what we did was to look how the maternal metabolism is influenced by different factors. So gestational weight gain, stress, obesity, maternal ethnicity, or of course maternal diet. So we looked what is really influencing the maternal metabolism in pregnancy and how this may affect offspring adiposity. So this is the first result slide, so I will take a few minutes here to explain it to you. What you see here on the left side are these 6 different factors, exposures, which may affect your metabolite. So we put all of these exposures in one single linear model, and then we got a p-value, like a p-value we got for a t-test. And you know a p-value, if it's very, very low p-value means highly significant association between, for instance, gestational weight gain and the metabolite. The problem in plotting low values is that you cannot plot low values. It's very difficult. So the idea was, and I think it comes from the genomics part, to use a negative log transform p-value. So if you log transform your p-value and use the negative value of this, you get a very high value of this for a low p-value. That means high negative log transform p-values stand for a high association of exposures to the metabolite. So when you, for instance, look here at the gestational weight gain plot, we have here the different metabolites. So I'm not sure if you can see it, but there are lines here, and each line presents a metabolite. I just mentioned here the group, so this is non-esterified fatty acids. And we plotted here the log transform p-value for each metabolite. So if it's in the outer part of this circle, it's a high association, because high log transform p-values present a high association. And in the middle, there's 0, and 0 log transform p-value is no association, would be a p-value of 1. And then we did it for all the p-values, for all metabolites, and all the exposures we got, and plotted all the 6 different exposures. And what you see here is that you find a lot of peaks for high association between pre-pregnancy BMI and different metabolites. But for instance, for the diet parameters, HEIP, which is a marker for the quality of the diet, and for total energy intake, which is a marker of quantity of the diet, there's less association. And even for gestational weight gain, there's just a few association, less association for sex, and just some for ethnicity. So you see at this slide that these different factors, diet, gestational weight gain, age, ethnicity, and pre-pregnancy BMI have different effects on the metabolome, and that pre-pregnancy BMI is the strongest predictor for the metabolome in pregnancy. So for me, this was very surprising, because I always thought these are the determinants for the metabolome. And pre-pregnancy BMI to be the most significant or most strongly associated exposure to the metabolome in pregnancy is some kind of a problem when you think of how to tackle this obesity pandemic, because you don't know if a woman wants to get pregnant in 2 years or something like this, but the state of this woman, if she's obese, still has an effect on the metabolome in pregnancy. What I forgot to mention is that we have these 3 different trimesters. So blue is the first trimester, red is the second trimester, and green is the last trimester. So you see there's also a different effect in the different trimester of the obesity of the women on the metabolism. So, yeah, maternal stress I did not show because it's quite complicated. There was no effect, and not even on any metabolite. Less associations for diet, weight gain, some of ethnicity, but the strongest predictor is maternal obesity. So this is a state of preconception and not in pregnancy. So what we do next was to look at which metabolites are affected by the obesity state of the moms. And you see here, there are a lot of peaks at this part for the non-esterified fatty acids. In the second trimester, so red was the second trimester. And this again is the same plot, just as a bar plot. So we have here the log transform p-value, and anything what is above means high association. And you see here that these light gray bars are really high, so there's a significant association, especially in the second trimester. So why is there a high association between obesity, adiposity, and free fatty acid in the second trimester? So non-esterified fatty acids are usually lipolyzed from the adipose tissue in the fasted state. That means if you're starving or you're fasted, you have no glucose, you have nothing to eat, so the body needs something for energy provision, and he uses the non-esterified fatty acids from your adipose tissue. So they are lipolyzed in the adipose tissue. Non-esterified fatty acids are transported to the different tissues, liver, muscle, or whatever, and there they are used for beta oxidation, to provide energy in this tissue, or in liver even for ketogenesis, so you have ketone bodies, which can be used by the brain for energy provision. So this means that the NEFA level usually depends on the adipose tissue. And this makes pretty sense with our results. Because in pregnancy, the first 2 trimesters, usually women gain fat, so they get more obese. Even lean women gain more fat than obese women, because they have to spare this fat for the last trimester, when the fetus is really growing, to have all this fat that the fetus grows. And so if you have a lean woman, of course she needs to storage a lot of fat. When you have obese women, she already has a lot of fat, and she does not need to store any more fat. And so the NEFA levels are higher in the obese women because they just don't have to store fat. The lean women store fat, so the NEFA levels go down because they don't do this lipolysis anymore, because they want to store fat. And so this makes pretty sense that the NEFAs are especially associated to the obesity state in the second trimester, where this storage of lipids is very increased in the lean women. Of course, obesity is also related to insulin resistance, which is also related to NEFAs, and could be another explanation. But what is also striking is that not all NEFAs are affected in the same way. So the free fatty acids, the NEFAs, it's the same. You have here the fatty acids with 1 double bond, the MUFAs, monounsaturated fatty acids, which are all very high associated to pre-pregnancy BMI. Then you have the omega-6 presenting fatty acids. So omega-6 are always the bad fatty acids somehow in the literature. So 23, 24, and 22,4, which are even in the last trimester associated to pre-pregnancy BMI. And what's more surprisingly is that the omega-3, the good fatty acids, have no association to pre-pregnancy BMI at all. So the monounsaturated fatty acids, 16,1, 17,1, 18,1, are elevated due to pre-pregnancy BMI. And even the ratio between the monounsaturated fatty acids and the saturated fatty acids is strictly associated with the obesity state of the mother. And this is because of one enzyme. It's called stearyl-CoA desaturase 1. And this creates, out of your saturated fatty acids you take in, you eat, the monounsaturated fatty acids. And they even found that an elevated SCD1 activity is linked to a switch in fat metabolism, more to an anabolic state. That means if you have a monounsaturated fatty acid, it's not that easily oxidized like a saturated fatty acids. Because it's not oxidized, it's stored in the fat, and you get more obese. And the other issue I already mentioned is the omega-6 and omega-3 fatty acids. There's a lot of literature available on omega-6 and omega-3 fatty acids. There's a lot of confusion available also, so nobody really knows what's going on. And different directions of the associations, so sometimes they are good, sometimes omega-6 are bad. This is really weird. But what's in our studies is now tackling is that the 23 omega-6 fatty acid called D-homogamma-linoleic acid is popping up more and more to be related to obesity development. So usually arachidonic acid, so 24, was the bad N-6 fatty acid. But now it's again the D-homogamma-linoleic acid which is popping up and was of course related in previous studies to obesity. So we now see that pre-pregnancy BMI has the strongest effect on the metabolome of the mothers, and it's especially the non-acidified fatty acids, the monounsaturated fatty acids, and 23 fatty acids. So the question now, what of these changes in the metabolome due to the obesity of the mother is really affecting the obesity of the child? And what we did was to look at the birth weight. So we had the birth weight percentile. It means it's adjusted for gestational age and sex of the infant. And then we looked how the metabolite may be associated to the birth weight. And we again found a wide variation here for the NEFAs to be related to birth weight. So the association was a little bit weaker, of course, because it's a little more far away from the mother. But again, the NEFAs were popping up, so that means that NEFAs, which are affected by the obesity state of the mother before pregnancy, are also affecting the birth weight of the children, and so we have to be very cautious about the preconceptional state of all these women and to look at the NEFA levels. What we also found was that lysophosphatidylcholines, a certain kind of lipids, were negative associated, but just in the lysophosphatidylcholines of the last trimester. What you have to be aware of, we just measured birth weight. So this is no marker for adiposity of the children. You have to do DEXA scans, skin folds measurements, so a lot of measurements to really analyze how much adipose tissue is in the children. So what's next? Now we have looked at pregnancy. Of course, we have also a lot of data of birth. So we got a lot of data of cord blood, which is an issue on its own, where we can look how cord blood is related to birth weight. And we did this in the LISA cohort. This is Elisabeth Thiering from the Helmholtz Institute in Munich, who did all the statistical analysis. And we got over 700 cord blood samples from a very healthy cohort to look how nutritional milieu, but there was not so much data, of the cord blood is associated to the growth and to the birth weight of the offspring. And what we found was a striking association of lysophosphatidylcholines to birth weight. So as I mentioned a few slides before, we found lower levels of lysophosphatidylcholine to be associated to birth weight in the mother. So that means that we have higher level in the cord blood, so on the fetal side, which are associated to birth weight, and we have lower levels of lysophosphatidylcholines associated to the birth weight on the maternal side. And what you see here again is that you have here the monounsaturated fatty acids in this lysophosphatidylcholine, and again you have the 23. And you have non-acidified fatty acids. This time you have the omega-3 presenting non-acidified fatty acids, which show no association in pregnancy metabolism at all, to be a positive predictor for birth weight. So what about the LPCs and birth weight? So this is from 2010 from Haggerty. So it's now 6-year-old idea how lipids are transported from the mother to the fetus. So here we have the maternal side. We have the lipoproteins, we have the free fatty acids, And usually the lipoproteins are broken down and everything is at the end of free fatty acids and non-esterified fatty acids, which is taken up by the placenta. And afterwards, there's some lipid metabolism in the placenta, of course. Again, free fatty acids are released by the placenta in the fetal circulation. So free fatty acids present somehow the transport mechanism, how to transport fatty acids from the maternal side to the fetal side. This was 6 years ago. And now there's a question, what about the LPCs? Since we found this lower level of LPCs in the mother related to birth weight and higher levels of LPC related to birth weight in the fetus. And so there's now a new paper published, just forgot to mention it here on this slide, showing that LPC can be uptaken by the placenta. And so the next step now is to look if LPCs in the placenta can be secreted in the fetal circulation. So this was some kind of hypothesis generating study, and now we have to cooperate with partners with animal models or just get some placentas and cord blood to look about the mechanism. But the role of LPCs, so even if you say, okay, LPC are transported via placenta to the fetus, it still needs to be explored. Even in adults, we did not really know what they do. There are some reports about higher levels in obese men, lower levels in obese non-diabetics, even the higher and lower levels depending on the different LPC species, so which fatty acid is in this LPC. And so there's a lot of confusion now going on about LPCs, but I can mention this because I don't have a slide because of the time issue, that we found LPCs always to be associated with obesity and insulin resistance in all our trials. But even we found higher and lower levels, so this really needs more investigations and mechanistic studies. And again, we have this popping up of these monounsaturated fatty acids and the 23 fatty acids and a positive effect or negative association to birth weight of the omega-3 fatty acids. And again, we also looked at the ratio of the monounsaturated fatty acids to the saturated fatty acids, and we found the stearoyl-CoA desaturase again to be somehow related to the birth weight. So this were pregnancy, cord, and now we look at infancy. So this was our usual first trial, was the SHOP cohort. We did in our metabolomics pathway work. This is Franka Kirschberg from our division. She's a statistician, so work with her sometimes is hard because she has a lot of ideas what to do and what not to do especially. And so we analyzed the SHOP trial. The SHOP trial was set up by Bertolt Koletzko now 12 years ago. And there we looked, or he looked, especially if lower protein formula has an effect on obesity risk in childhood compared to higher protein formula, which was given to infants in the first year of life. So there was also a breastfeed group, but you can just use breastfeeder as an observational group because you cannot randomize this both group. You can force nobody who wants to breastfeed in an intervention group. So we analyzed blood samples at 6 months of age, and from each group we got around 200 up to 300 samples. And what we found is somehow an acknowledgement of the high protein hypothesis set up by Bert Koletzko. So that high protein intake during the first year of life was elevated to higher insulin and IGF-1 levels, but it was also related to an increased weight gain in the first months of life. And this is a paper from our group, from Martina Weber in 2014, even at school age. So this is 6 years. The high protein group had a higher risk for obesity, although they just received the different food here in the first period. So this is where they received the high protein diet. Afterwards, everybody eats the same nearly, but still there was a higher prevalence of obesity or of overweight, I think. Here we have overweight in the group which received in the first year of life this high protein. So okay, we could show these ideas may be working, but what is the mechanism behind this? So we just applied our metabolomics platform at samples from 6 months of age, so when there was still the intervention going on in the children, and we found that the amino acids, this time it's amino acids, were elevated in children which get the high protein formula. So this is again the log transform p-value, and everything what is above this dotted line is higher in the high protein group, and anything what is here is higher in the low protein group. Of course, if you take in more protein by higher protein formula, you got more amino acids because proteins are kind of built up of amino acids, and especially the essential amino acids are affected. That means for the non-essential amino acids, the body somehow can regulate the metabolism. But what we found for striking was that the branched-chain amino acids were partly more strikingly associated or higher in the high protein group. And this is for just a simple reason. When we looked in the literature, we found that there's a first-pass metabolism of amino acids. It's just like other first-pass metabolism. So we have the dietary intake, then it's getting absorbed by the portal vein and first transported to the liver before it even enters any other systemic circulation. And what we found is that the key enzyme of the branched-chain amino acid is missing in the liver. So all amino acids are somehow transformed, metabolized in the liver to whatever, except the branched-chain amino acids. So they are just released the same way they got into the liver, in the systemic circulation, and so they have a higher association to the dietary protein intake. In this circulation, they are transported mostly to the skeletal muscle, where they are used for branched-chain acid oxidation. And the products of the branched-chain acid oxidation are short-chain acylcarnitines, which were also elevated in the high protein group. Of course, it makes pretty sense. We have a high protein intake, higher amino acids, higher branched-chain amino acids, and when they are oxidized, we get higher acylcarnitines, which are the products of this branched-chain amino acids. So this is a metabolism, and the key enzyme of this degradation of the branched-chain amino acids to this acyl change is a branched-chain keto acid dehydrogenase, which is at this step. And so we looked how, if there's maybe a different association of the substrates, this branched-chain amino acids, to the different acylcarnitines. And what we found that in physiological levels, which is here, there's a normal linear increase. If you have higher branched-chain amino acid levels, you get higher products. But at a certain point, we call it a break point, you find, especially for leucine and isoleucine, you find that there's some kind of saturation. So we did this correlation model, and we found here this linear increase. And at this point, some kind of decrease or plateau established. And we concluded somehow that because of this higher protein intake, you get too high branched-chain amino acids levels, and so your degradation pathway gets somehow saturated, and the BCAA can exceed their own degradation, their own regulation. And BCAAs are known in the metabolomics world a lot to be associated with obesity, and this is for some reasons. So usually BCAAs are not metabolized in the liver and secreted by the liver just from the diet directly in the systemic circulation, because they are a signal for protein intake. That means if you have higher protein intake, you get higher branched-chain amino acids levels, and then, of course, you get a signal to the tissues that they can create now protein, because they have a lot of amino acids now available to create protein and stop protein breakdown. They also have an effect on insulin, which supports protein synthesis. But branched-chain amino acids also have an effect on the fat metabolism. So you see that leucine is slowing down beta oxidation, but it's increasing lipogenesis. So if you have higher protein intake, fat is no longer oxidized, it's stored in the adipose tissue. And this may be a pathway how this elevated branched-chain amino acids, which escape their own degradation pathway in high protein intake, may have its adverse effects. And so we looked at the ratio of long-chain acylcarnitines to free carnitines. As I mentioned on the non-acidified fatty acid slide, acylcarnitines are these metabolites which are involved in the beta oxidation, the fat oxidation. And when you create this ratio here, this is a marker for carnitine palmine transferase, the first step of beta oxidation. And in the high protein group, we found this marker lower. So we really could conclude that there's lower beta oxidation in infants which received this high protein. And maybe this is why you have a higher fat storage in adipose tissue and have a higher obesity risk later on. So I've harmed you now with a lot of these things and results. And of course, we also have a lot of studies going on in childhood and adulthood where we look if changes at this period or exposures at this period have already affected childhood. Just because of time reasons, I will skip this part, but I can just say, what we will do now. We have all these trials I already mentioned. We have the NIFAs, which maybe play a role in pregnancy and early infancy. Amino acids may more play a role in development of obesity and diabetes in the later life. So we found in a trial at 10 years association of aromatic amino acids to insulin resistance. At adulthood, early adulthood, we found sphingomyelins and lysophosphatidylcholine, so lipid to be associated with different styles of insulin resistance and adiposity. We have the phospholipids, especially the LPCs, which play a role in this transgenerational circle maybe of obesity, but they are also all related in all these studies. And we have these 23, which is also popping up in all these studies. And the branch chain I already mentioned in aromatic amino acids. We also have the non-saturated fatty acids, which are also somehow linked to each state of obesity and insulin resistance. But what do we have to do now is to repeat all this analysis. I mentioned we just measure humans, so we just measure plasma. So we just can do association studies. It's very difficult to get any conclusion about this, about causality or even mechanisms. And to be really sure, you have to repeat analysis in different cohorts, or just do like in the CHOP trial, do longitudinal follow-up. We have more and more pregnancy cohorts where we just can repeat the measurements we found in the UCI cohort. And this is the only way you can create evidence in human cohorts in the plasma samples, because you cannot do so much more. And what we also do now is to look at different omics techniques. So I have just presented results from the Metabolomics platform we have now, but we're now just setting up trials where we look at epigenetics, proteomics, and metabolomics in one kind, because it's somehow playing a role all together. So epigenetics highlighted, and this is a slide from Richard Severy, with whom we also have some cooperations. And this shows that you have these exposures, for instance, maternal diet, or you can also say maternal obesity. This is maybe disrupting the epigenetic profile, then has an effect on the metabolome, and then is associated to the adverse outcome like birth weight or cardiovascular disease. So to look really at the mechanistic of all this, you really have to look at epigenetics. Maybe you have to use genetics, and of course metabolomics and transcriptomics and genomics data. Though, of course, there's a logical conclusion that there's an exposure effect in the epigenetics, which then affects the metabolome. But it's, of course, also vice versa. If you think of epigenetics and of methylation pathways, metabolites are, of course, involved in all this one-carbon metabolism. So this is, again, a slide from Richard. And you see here a very complicated pathway of all the methylation process. Where is SAM? Here's SAM, the most famous methylation metabolite. And these are all metabolites we can measure with our platform. So metabolites we can measure are somehow associated to epigenetic profiles. And so the diet, so this is what here, the early nutrition, if you have folate in your diet, if you have choline in your diet, betaine, homocysteine, and methionine, so amino acids, they may affect the different metabolites in the cells. And so they can maybe also affect the epigenome. And this was shown by Waterland, that there really is an effect of different amino acid cultures to different cell cultures, media to different cell cultures. But there's also some trial which showed that low protein intake in pregnancy. So low protein intake is associated with lower amino acid intake, so we get lower amino acids. And there was a lesser epigenetic profile for some metabolites which are in enzymes, or genes of enzymes which are involved in lipid metabolism. So this is my last slide, and to come back to the topic of this talk, metabolomic and epigenetic mechanism, the key. So it's very hard to say, of course, because you have to create all this evidence. As I already mentioned, it's very, very difficult to really get evidence. I think there's a lot of going on in human studies right now, looking at early programming and epigenetics, and they found different results. And so it's very hard for them to conclude which is the right way. And so you have to do integrative omic approaches just to study the complete mechanism. But for evidence in humans, as I said, you have replications of findings. You need large longitudinal follow-up studies just to see if there's a causality between epigenetics, elevation of metabolites, and later health outcome. And of course, you need large ones because you have a really high dropout in children, childhood, adulthood, puberty. You know, the girls and boys just don't want to go to the follow-up of your study. And so you really need large longitudinal cohorts, and then you have to repeat it, so it's very expensive. And the problems in humans, of course, is that you have environmental factors, you have genetic factors, and it's not a controlled environment. And you always have to mention this in your trials, and this is very hard to set up a life course trial and even repeat the same life course trial with the same factors. So I have to thank, of course, the team. This is a famous three of the Metabolomics in LMU. Olaf was the lab, Franka was doing the statistics, and me was talking, and all the other guys, Bert again, who is our supervisor, and getting all the fundings by these different European projects, since it's a very expensive business to do metabolomics. And of course, thank you all for your attention, and open for questions now.
Very interesting talk. My question will be the following. What's your take on the supplementary diet for athletic performance using BCAA, L-carnitine, especially in the long-term effect of what this is causing?
Yes, this is a very interesting question. So I'm not really a fan of BCAAs, but there are a lot of studies showing the relation of BCAAs to diabetes and obesity. But on the other hand, there are studies showing that physical activity and BCAAs are also related, since a lot of sportsmen use BCAAs to create muscle tissue. For the longitudinal follow-up, there's one study, I'm not sure who wrote it, which showed that there's an alteration in the metabolism of amino acids 10 years before onset of insulin resistance. So they could show, with samples they collected 10 years before, that you can predict later onset of diabetes and insulin resistance even better compared to glucose and insulin at these samples at 10 years before. But when it comes to the so cross-sectional, where they looked if, okay, now you have diabetes, there was no association between amino acids and so on. So my opinion is there is something going on. Maybe amino acids are affected a little bit earlier than glucose metabolisms. So you can use it as predictive effect, but you have to be very cautious about this, and you really have to repeat it because there's different literature available. I hope this fits your questions.
What are your thoughts concerning the transgenerational aspects?
So,
Suppose you have an idea.
sorry?
What are your thoughts concerning the transgenerational aspects of this environmental influence and the cycling? Because I assume that you have been very careful to say that there wasn't a cycle.
Yeah,
and of course, I don't want to say that this is transgenerational. It's not in any case. But what are your thoughts concerning that? And how is this nutritional environment influencing the epigenome?
Yeah, thanks. It's a very good question about this intergenerational cycle. It's very hard because you have the genes in the women and in the children. Maybe it's the genes which is setting this. Of course, you always have a not so good, a bad diet by the mothers who are obese because they're grown up like this and they are just giving it to their children. So it's not a molecular mechanism, it's just a dietary mechanism. Yeah, so my idea is that when you think probably there is a mechanism, when you have to speculate about a mechanism, my idea is that this maybe the omega-6 fatty acids, because they have a higher dietary intake in the obese women, and there are some trials showing that the numbers of adipocytes can be regulated by the different omega-6 fatty acids. So if you have an higher omega-6 fatty acid intake in the mother, the children have more adipocytes. And then the adipocyte number is fixed, this is known, and then they have more adipocytes and then can more get obese, but my main idea is it's diet and not this mechanism.
Okay, here. Yes, I have a question, sorry. You didn't find any relation between the mother's diet and the metabolites you measured, but there might be a relationship between what the mother eats and the baby. And so which metabolites must be measured to answer that question?
So if you say, okay, we didn't find any association between diet and metabolites, maybe we measured the wrong metabolites. What we missed here were the triacylglycerols, so the lipoprotein content of the mothers, where maybe most of the dietary fat intake is stored. But since this dietary assessment was for a longer period, I'm really surprised by these results, so I cannot explain them. I really expected that there's big association between fat intake and lipids in the mothers. Maybe we just missed the triacylglycerols. We should look at this. Otherwise, I have just no idea. But this is a really good question. Yeah.
Bueno, agradecemos al Dr. Helmut por su presentación.
Thank you.
Muchas gracias.
Thanks.
Y bueno, los esperamos mañana temprano. Por favor, sean puntuales porque la puerta se cierra en cuanto empiecen las sesiones. Gracias. Thank you. Thank you very much.
Yeah, yeah, I hope it was clear.
Is it possible to design the target program or something like that to avoid all these shapes?
So you mean if I have all data in one?
Yeah, I
So
mean,
we-
they are multifactorial.
Yeah, they're multifactorial, you have all these cohorts, so we have to do it because we have a project where
Do
we...
you know what I'm interested
Yeah.
with my child, I have insulin resistance
Ah.
and I am worried because of my response.
Ja, it's very, man kann es irgendwie alles zusammen machen, aber es ist sehr schwierig, because auch die verschiedenen Studien haben alle ein verschiedenes Grundgerüst. Ja, das sind alle nicht so gleich. And it's very difficult. But we have to do it, and because we have this one EU project which is finishing now, and the last step is to do an integrative approach.
Yeah, and also
I have no idea.
the same people.
Yes, this is very difficult, but we try it. Yeah, the follow-up, we try it, and all the studies we have. But
And you cannot control what they are eating or if they are really
yeah, you just can recall it, but you don't know the complete take. Oh, yeah, don't worry. Thanks a lot. Yes, and I just have to give someone my